So now we're going to learn about transcription. Transcription is the process where the DNA molecule, which is our parent strands of DNA, are copied or rewritten onto mRNA. So mRNA is the messenger, and we're going to see this process of copying a piece or a portion of the DNA molecule over to a messenger RNA. And it works very similar to DNA replication. The first step is DNA is unzipped by helicase. Remember, DNA is a double helix, and it is stuck within the nucleus. So once the bases are exposed, the complementary bases will attach to the exposed bases on DNA. The major difference between DNA replication and transcription is wherever we would see a thymine, we are now going to transcribe a uracil. So adenine will pair with uracil during protein synthesis or the process of transcription. So the completed messenger RNA detaches from the DNA template, so that means it leaves the parent strand, and then leaves the nucleus through a nuclear pore. Once the messenger RNA leaves the nucleus and is in the cytoplasm, it will attach to several ribosomes. Ribosomes are the site for protein synthesis, so we will be making proteins. So for every 3 bases on the mRNA, that corresponds to a particular amino acid, and we'll call this a codon. So this diagram is showing you the double helix of DNA, and that's going to be in red. That's the first step. The second step is going to be to make mRNA, which is the messenger that brings the message, so it rewrites the message first in the nucleus, and then brings that message to the ribosomes to make proteins. So that's the process of transcription. The next step is translation. So when we're translating the message from messenger RNA to a protein or an amino acid. The amino acids are delivered to the correct codon on the messenger RNA by transfer RNA or tRNA. Transfer RNA are like taxis that deliver amino acids. So tRNA have anticodons that correspond to the codon on the mRNA. So the amino acids are delivered to the correct location. So for every 3 bases, that's called a codon on mRNA, tRNA will bring those complementary bases, which is the anticodon, and the tRNA brings a specific amino acid. So what this looks like, tRNA, we have our amino acid at the top, and then we have the anticodon. Those are the 3 bases that will match or they will correspond to mRNA. So here's the full process. We have DNA stuck inside of the nucleus with its specific blueprint or codes. mRNA copies the specific code that needs to be made. mRNA leaves the nucleus and goes into the cytoplasm to the ribosomes. Once the ribosome attaches to the mRNA, it's almost as if we're feeding tickets into a ticket counter. And the mRNA is counted. So for every 3 bases, the codon, we will see the tRNA bring over the anticodon with the amino acid attached to create our polypeptide chain, which has to do with our proteins. So our base pair rules for DNA, when we transcribe, so we're writing the message on mRNA, we'll see uracil. When we translate the message back to tRNA for the amino acid, it will be the complementary base pair. So mRNA brings the complementary bases to tRNA, and then tRNA matches the complementary bases from mRNA. So you'll see a major similarity between DNA and tRNA. The major difference we see between the two is instead of carrying uracil on DNA, we see thymine. Instead of carrying thymine on RNA, we have uracil. So this is the process of protein synthesis. We first see transcription, then we're going to see translation. Now let's look at the secret code, which is our amino acids and codons. This chart is used only for mRNA. So we have our first base to the left, our second base on the top, and our third base to the right. Gene expression practice questions, part 2. So we have transcribe the following DNA sequence to mRNA. So we are given the DNA sequence. We have to write the mRNA sequence. If you look to the right, you see DNA and RNA, a key for you. So T will pair with A, G will pair with C, C will pair with G. Now, the next we have adenine. Adenine will pair with uracil. That's going to be the difference between DNA and RNA. G will pair with C, so guanine with cytosine. T will pair with A. And finally, we have T pairing with A, so thymine pairs with adenine, cytosine pairs with guanine, and adenine pairs with uracil because we are talking about RNA. So how many codons are there on the DNA strand above? Well, we drew lines for you to separate the 3 bases into codons. So we have 3 codons present in our mRNA above. Now, looking at our previous table, we have to use the mRNA to figure out what amino acid the mRNA corresponds to. So first, the mRNA we're looking at, the first codon, is ACG. Let's go back to our previous slide and look for ACG. So we look for our first base, A, our second base, C, and our third base, G, and I've circled them for you. Next, let's see where we need to go. So we follow the C down to the A row, and then we follow the A to the C column. Now we're in one box that all of the arrows are pointing to. So the G will also have to follow the arrow into the C column. So we'll follow that over, and we'll see ACG. That was our mRNA message, our codon. So our amino acid is THR. Let's see if we're correct. So THR. We're going to do that again for UCA, and again for our last codon, AGU. Go ahead and give that a try and then check your answers. So if you received SER for your second codon on mRNA, great job. And SER for your third codon on mRNA, again, wonderful job. If not, please continue to practice. Next question, what are the 2 main stages of gene expression? So that's transcription, where we write the message, and translation, where we change the language from nitrogenous base pair to amino acid. Number 5, what is made when genes are expressed? Proteins. Remember, long chains of amino acids make polypeptides and therefore make proteins. And finally, question number 6, what molecule acts as a template in gene expression? DNA. Remember, DNA is where the message is copied. mRNA copies the message, so transcribes the message with the complementary bases for RNA, leaves the nucleus, brings the message or the codons to the ribosomes, and at the ribosomes, tRNA brings over the amino acids for proteins to be made. Next, we're going to use what we've just learned to answer the questions with the diagram. So base your answers to the following questions on the diagram of a part of a cell and your knowledge of biology. What kind of molecule is being formed? Remember, the molecule that we're forming is a protein. So we see structure X, we have to identify. That is called a ribosome. mRNA brings the message from the DNA molecule that's stuck inside of the nucleus to the ribosome for proteins to be made. C, what structural evidence indicates that the labeled strand is mRNA? Remember, uracil is present. DNA does not have uracil. It has thymine. So let's go over those bases in the pairing rules. So for DNA, adenine pairs with thymine. Guanine pairs with cytosine, and vice versa. So it can go backwards as well. So cytosine can pair with guanine, and thymine can pair with adenine. For RNA, adenine pairs with uracil. Cytosine still pairs with guanine, and again, we can do that in the reverse. Guanine pairs with cytosine, and uracil on our mRNA will pair with adenine on our tRNA. Remember, mRNA is also single-stranded. What amino acid will connect with the next codon, GAU, on the mRNA? Remember, you have to use your mRNA codon chart, which is for amino acids. And if you were able to figure out ASP, which is a type of amino acid, great job. Test yourself, part 2. So if you're given DNA is in the nucleus, it contains the codes or the recipes needed to make a protein. DNA is like the reference book in a library that contains recipes. Just as the reference book cannot leave the library, DNA cannot leave the nucleus. If you need to bring the recipe home so that you can use it, you must first photocopy the necessary pages from the reference book. In a similar way, if the recipe for a protein is needed in the cytoplasm, it must first be copied onto messenger RNA, which is mRNA. That's transcription. Just as you leave the library and bring your photocopy of the recipe home, the messenger molecule leaves the nucleus and goes to the cytoplasm. When you get home, you put your photocopy on the kitchen counter so that you can read it. In a similar way, the messenger molecule attaches to the ribosome so that it can be read. Just as you might have helpers to get the ingredients needed for your recipe and bring them to you, there are special transfer molecules, or tRNA, in the cytoplasm that deliver the amino acids needed to make particular proteins to the ribosome. So let's do a little review together. So what is the reference book or the cookbook in the library similar to? DNA. What is the library similar to? The nucleus, because our recipe book is stuck inside of the library. The photocopy of the page containing the cake recipe you want to use is mRNA. Remember, it's a photocopy. We transcribed it. Your kitchen counter where you're going to follow the recipe to make the cake? The ribosome. The ribosomes are going to make the proteins. The helper who goes and gets the ingredients you need is the tRNA. Remember, they bring over the amino acids, so the T codons. And the cake is our protein. Let's label the diagram. So, the nucleus is going to be A. What's leaving the nucleus? mRNA. Remember, DNA is stuck inside of the nucleus. We have the ribosome. Remember, the mRNA brings over the message to the ribosome. Then we have tRNA bringing over the T codon, or the amino acid sequence. The T codon is what's attached to tRNA. And remember, the codon is what's attached to the mRNA. Finally, we have the amino acid that is brought over by tRNA because of the anticodon. So what is protein synthesis? Well, synthesis means to make, so we're making proteins. It's also referred to as gene expression. The 3 types of RNA involved are mRNA, tRNA, and ribosomal, or rRNA. What is the function of tRNA? It's going to bring over the amino acids because it's going to have the T codon that will be the complementary base pairs from the codon on mRNA. So this occurs in 2 phases. We have transcription and translation. Transcription occurs in the nucleus and produces the mRNA strand that leaves the nucleus and goes to the ribosome, which is in the cytoplasm. The ribosome will produce the proteins.